What happens when you combine imagination and science? Abdul Hadi, our next TEDx speaker.
So, I'm quite humbled to be here today. I won't have very much in terms of images at the beginning, but there will be some pretty cool photos later on. Yeah, so I'm going to talk to you about some funky science. I can see that some people here might be a little bit afraid of science, but I can tell you I'm more afraid of you than you are of me, because it's actually very difficult to tell people about science, particularly if they're not a scientific audience. So basically, I'm coming to the UAE to shamelessly promote the idea of biophotonics. So what is biophotonics? Most of my friends are not scientists, and when they usually ask me what biophotonics is, I get them to guess. And one of the cleverest guesses I had was that biophotonics is photography of animals. I will show a photo of a jellyfish later in the presentation, but generally speaking, biophotonics is not the photographing of animals. And I'm going to avoid entirely all equations. So, biophotonics is the clearest example of why there is such a thing as the natural sciences, because biophotonics is basically a conflation of physics and, in fact, sort of advanced quantum physics, 20th century physics, in a sense, and cellular biology, which are, in the past, have been at two opposite ends of the spectrum of science. And in pictures, in the past, there was this great saying by Niels Bohr, who was one of the original founders of sort of atomic theory, who said that in science there is only physics and the rest is stamp collecting. But with biophotonics, we actually have an image of science as a whole, because we're able to make quantitative, exact assessments of what actually goes on at a cellular biological level, which is actually much more difficult to do without biophotonics. So in a sense, biophotonics is about providing certainty by utilizing light and understanding exactly what happens at a cellular level and at a subcellular level. You have to remember that a biological cell, and whether that's the biological cell in any of our bodies, or the biological cells in mice, or the biological cells in trees, are cells which are typically a few micrometers across. We measure them in micrometers, and a micrometer is about a millionth of a meter. Whereas some proteins that you might care to really image are at the nanometer level. Well, there might be several hundreds of nanometers or several tens of nanometers in some instances. In some instances, you're looking for a molecular interaction which is occurring at sort of, yeah, at the scale of a nanometer, so 1,000th the size. Typical optical microscopes don't allow you to image with that kind of resolution. But what biophotonics does is that it allows you to, let me just make sure I'm on the right slide. It allows you to pinpoint those molecules within the cell with certainty by sort of activating them. And you activate them with these things called fluorophores. So a fluorophore is a special molecule which absorbs light in one particular frequency. You might remember that light of different colors is basically light of different frequencies. So blue light has a higher frequency and a higher energy than green light. So these fluorophores will absorb blue or ultraviolet, and then they'll emit in green. They're fluorescent green. At least my favorite fluorophore is the green fluorescent protein, the GFP. And for some reason, it always reminds me of the BFG, the big friendly giant, but it's somewhat different from that. Their activation, by activating them and getting them to fluorescent green, you can pinpoint things at 1,000th the time the magnification that you would with a typical optical microscope. Oh, there was a slight mistake here, because anyway, it all started with the green fluorescent protein, which was extracted from these jellyfish. They had to kill something like several tens of thousands of these jellyfish to extract enough protein, because they produced them naturally. So they extracted them and then found ways to synthetically create more of these proteins, which they could then splice into animals or like plants, for example. You might have seen, has anyone here seen images of the green fluorescent cats on the internet? You've seen these, okay, so at least one person has seen the green fluorescent cats. You can Google like, fluorescent cats, and basically what they've done is that they've genetically mutated them so that they express this protein, and you then shine sort of an ultraviolet light on your cat. This is not something I encourage you to do at home. And the cat will then glow in green. And the point is, if you can tag this in such a way, let me, before I get to that, if you can tag it in such a way that the protein is expressed in a particular way, what you can do is you can follow the progress of a virus throughout the moving through a cell, or you can follow the progress of, let's say, a growth hormone or something by genetically mutating that virus or that hormone, and then watching it glow in green as you flash ultraviolet light at your cell samples. And this is just an example of what I did. So again, there's something a bit strange with the slides, but we won't go to that. On the left is a cell, an antigen-presenting cell, which is basically, in this case, it's from the Epstein-Barr virus. And on the right is an MHC complex, which is from the human immune system. When we actually did these images in the lab, they were cleaned up a little bit more. But basically, what you find is they were tagged. I didn't do this myself, but they came to us tagged, and then we did the imaging. And you can see that around the edges of the cell on the left, there's like sort of a slightly brighter thing. And the reason for that is that it's fluorescing in green. You have to believe me when I say it's green, because it's a black and white camera, but we used a green filter to collect the light. So this is all the light that you see collected here, all the photons that you see collected here are green frequency photons. And what we were looking to find was how proteins, basically these protein bridges were built when the human immune system attacked cells from the Epstein-Barr virus. I did have images that other researchers have done as well. So you might think, so what? So you can look at all these like funky proteins. What does it mean for me? Why should I care? You should care basically because medicine is moving into a realm now where people are taking a much more, again, just as biology has moved into biophotonics and become much more exact, much more quantitative, medicine is also becoming much more precise, to misuse a scientific word in this case. It's becoming a lot more exact. People are interested in finding innovative ways of attacking disease, so not necessarily one vaccine fits all sizes for everyone, because you might find, if you use biophotonics, if you use fluorescence imaging, which is the kind of examples that I've been showing you, that different individuals have different processes or their immune systems react differently. You might find very specific ways of targeting a disease because you know a lot more about how that disease develops at the cellular level and at the subcellular level. So I guess the crucial word is tailoring or bespoke medicine. And why the UAE? I'm from the region, I'm from very near, and I feel very strongly about the fact that, sorely we're lagging behind. I could mention several papers, but if there's a particularly good paper which I found, which is not terribly specialist, accessible to everyone in the audience, is open source and can be found on Google Scholar, is by Hani bin Amr and Umran Bakush. Ironically, they're both in the UK, but they wrote a paper in Biomed Central about sort of the lagging behind that the Arab states have in terms of biomedical publishing. And I think that biophotonics is a particularly good idea for us to start investing in as a region, because it's still new enough. I mean, biophotonics really only sprung up in the 1970s, and it's still new enough that we can make really crucial advances. The technology can be bought into. I mean, the images that I showed, like right here, it took really just a few £1,000s to get the kind of equipment needed. I mean, there was some funky microscopes. There was a lot of effort that went in, of course, but it's new, it's inexpensive, it's interdisciplinary, it provides the sort of training that people are looking for and the kind of worldview that people in the 21st century need to advance. But anyway, if you look up the Ben Amr and Bakouche paper, it's from April of 2009. It's open source, so anyone can get it. It's slightly a depressing read, but the question is, the thing is, it should also be an impetus for us to kind of move a little bit forward. And I've realized I've probably said a little bit too much about this one. I also was particularly struck by the fact that I feel that the ethical traditions in our part of the world are not particularly averse to advances in biotechnology. And I know that, for example, in the UAE, they very valiantly went ahead and tried to, for example, tried to find ways of cloning camels. And I was struck by the fact that in this part of the world, our population didn't really kick up a fuss about that. Whereas, when Dolly the sheep was first cloned, there was a kind of sky was falling sort of mentality in a lot of countries that perhaps people had crossed boundaries. And I'd like to think that our own traditions, although we're clearly lagging behind right now, have sort of prepared us to accept innovation in science. And that brings us to I should say, I'm at least the third person today to mention Darul Hikmah. Now, if you're historically minded, you know that this place is not the original Darul Hikmah that was destroyed in the 13th century, but a new building that was set up a new complex that was set up on the same site in Baghdad. But my point is that our region in the past has thrown up great people like Hassan bin Thabit and Ibn al-Haytham. And if we make the right choices in terms of strategically encouraging science and nourishing new interdisciplinary strands of science like biophotonics, we can be great once again as we were in the past. There are a few words of caution. The images that I'd shown that I'd taken myself were quite humble, but it meant, even to get those, it meant sleeping in the lab. It meant, many nights of not being able to do other, enjoy hobbies that I would have otherwise done. Biophotonics and science in general, I think it's illustrative science in general, it takes a long time before the real benefits are reaped. So the green fluorescent protein was extracted about 30 years ago. 2 guys won the Nobel Prize for discovering it just last year. So it's about 30 years. It also doesn't, it's not going to produce immediate, quick rewards in terms of cash. And in fact, I mentioned that 2 of them won the Nobel Prize last year. One of the guys who discovered the green fluorescent protein, one of the guys who was involved, is a guy named Douglas Prasher. And as far as I know, I was hoping to verify this 100% before I came up here. But I'm quite sure that basically he now drives a van, a courtesy van for a hotel chain, instead of being like an academic scientist. So you can get completely sidelined by science. It's a very difficult thing. I don't want to discourage you from doing it. In fact, I'd like to encourage any young people who are considering science to give it a try. But the fact is that it's not immediately gratifying. But once you do get any results, as humble as they might be at the beginning, they're immensely pleasing. So it's about patience as well. And I want to thank you all for listening. I want to particularly thank all the people at TEDxDubai, Giorgio. Habiba Hamid pulled a great miracle last night by being able to get me into the country, so thanks, Habiba, as well as the people at Emirates Airways. And I particularly have to thank the King's College London Centre for Biophotonics. That's where I did my research. They're great people, especially Klaus Schuling. He was my research supervisor and a great guy, a great sense of humor amongst other things. And yeah, so look up the King's College London thing. If anything I said has been of interest to you, hopefully you'll look them up. And I can show you some more images because for some reason I think my PowerPoint presentation didn't work in the right sequence. But yeah, have a good evening and thanks for listening to me.
Fantastic. Thank you. Thank you. That was great.